Hello everyone, welcome back to another class on regeneration biology, and in today's class we will learn about limbal stem cells and artificial cornea. So we have discussed about limbal cells very casually in one of the earlier classes. In general, limbal cells are the transition zone between your cornea and the conjunctiva. So that is the transition zone, and the limbal stem cells play a major role in maintaining the transparency and the functionality of the cornea. So let us see. The cornea on the eye is a transparent structure that is present in the front of the front part of the eye, and it is our window to the whole world. You are seeing the world through the cornea. Although you can tell it is, I see the world through the eye. When you donate eye from cadavers, when you collect it, it's mainly the cornea. So because cornea can be transplanted without too much of immunological complication from any donor, and a recipient can have it for several years. So the maintenance of corneal tissue transparency is very important for the vision. So the integrity and the functionality of the outermost corneal layer, that is the epithelium, can play a key role in the refraction of the light onto the retina at the back of the eye. The thickness of the cornea can vary, like people who watch a lot of gadgets, and their cornea can get thicker and thicker, because of which it will act like a lens, which is the cause of your the short sight or the myopia, that the image will be formed in front of in front of the retina, not on the retina. So that is why doctors will give you a concave lens which will deflect the light, and so that the image will be formed, because it look like you have a lens in your eye, and in front of that there will be one more lens. So the thickness of the cornea is an important factor in deciding your eye power. So these days LASIK surgeries have come up where you can reduce the thickness of the cornea, and you can even get rid of your glasses. But the thing is, you need to have a good power of 8 or above, then the success is more. If you have a power of 2 or 3 or 4, maybe it is very difficult to get it done through the LASIK. I don't know, maybe technology may have improved now. So the integrity and functionality of the outermost corneal layer, the epithelium, is very, very important in projecting the image to the back of the eye. Similar to other epithelia, the epithelium of the cornea is maintained by the stem cells. So exploring the properties of these stem cells, along with the clinical consequence of the stem cell failure and the potential for stem cell therapy in ocular surface recognition, is a strong field of regenerative medicine, because there are n number of scenarios. Not that you have to get exposed to a acid or some chemical to damage your cornea. It can be mechanical injury also. Sometimes what happens, these limbal stem cells not doing their job adequately, because of which the cornea can lose its transparency, because there can be vasculature, etc. So there are multiple scenarios in which the cornea can lose its perfect way of transparent structure or perfect way of functioning. So limbal stem cells and the development of artificial corneas, if you look further, you can see the limbal cells or limbal stem cells and the development of artificial corneas are very much connected. They are related through their role in the corneal repair and regeneration. So your cornea it's a dynamic cornea, in the sense it constantly gets repaired, it constantly gets rejuvenated. That is why the thickness also is varying. So it's not that your cornea is a dead structure, just that it doesn't have blood vasculature. Limbal stem cells, short form known as LSCs, are crucial for maintaining a healthy corneal epithelium, and their deficiency can lead to significant vision loss in multiple ways. It can be simple peeling off of the epithelial cells because it's not getting replenished. There can be many such troubles can come due to the lack of adequate amount of LSCs. So research on LSCs is pushing the development of artificial corneas, which aim to provide a functional substitute for damaged corneas, and often using LSCs or their derived cells. Although we call it artificial cornea, it simply means it's not that it is made of plastic or rubber or something. It is made of cells only, but you collect these cells and tweak in vitro and expand in number and see whether can I fix the damaged cornea. Just kind of take the limbal cells and you culture them and then repopulate back. So limbal stem cells, they reside in the limbus at the border between the cornea and the sclera. That is the just like I told you, in your eye, if you look through the mirror, you see the black and the white. So the black portion is, we call it as a pupil, which is covered by the cornea, and adjacent to that, transitioning into the white, that middle portion, we have the limbal cells. They are responsible for regenerating and maintaining the corneal epithelium and the cornea, which is the cornea's outermost layer. Damage to the limbus or loss of LSCs due to various reasons. LSCs can be low in number, their life expectancy has reduced, etc. etc. So loss of LSCs can lead to limbal stem cell deficiency. It is known as LSCD, limbal stem cell deficiency, resulting in corneal conjunctivitization, which basically means the conjunctiva is walking into the cornea, which makes it opaque, and neovascularization and the vision loss. If the cornea is just like you close your eye, you don't see anything. It's nothing but you are cutting off the light. Same way, if your cornea became opaque because of the conjunctivitization is moving off the conjunctiva into the cornea, and naturally the blood vasculature will come and your vision will be lost. LSCs are being explored for their use in various corneal repair and regeneration strategies, including transplantation of LSCs or cells derived from them. Both ways one can approach. So how do we do artificial cornea development? The development of artificial corneas is driven by the need to treat LSCDs, that is corneal deficiency, limbal stem cell deficiency, because of which the corneal's structural deficiencies has to be met. And the other corneal disorders also can come in where the natural regeneration becomes impossible. That is why you have a trouble with the cornea's transparency or its translucency. So researchers are exploring different types of approaches, materials and approaches, including bioengineered scaffolds and cell-based therapies using the limbal stem cell and their derived cells. You can have the limbal cells, culture them, and you expand in number and then try to put it back. So some approaches focus on creating a corneal substitute that can be implanted to restore the vision and maintain the corneal clarity. So this is the approach what is usually done. LSCs and their derived cells are being investigated for their potential to repopulate and maintain the artificial corneal surface, mimicking the natural corneal epithelium. Slight changes in the corneal dynamics, etc., can be adjusted using the correction glass. Not that everyone who have got a cornea, thickened cornea, they have to get an artificial cornea. They can simply do a correcting reading glass, can be fixed. But some rare cases where the cornea, although it is thick, it is not transparent, it is not biologically usable cornea. That is the situation the cornea fixing is needed. In summary, the limbal stem cells are essential for corneal health and regeneration, and their study or the working of or the exploration of the limbal stem cell is crucial for advancing the development of artificial corneas. Artificial cornea development aims to provide a functional substitute for the damaged cornea, often by using the limbal stem cells itself or their derived cells to create self-renewing and transparent corneal surface. So when you make this limbal stem cells expand in number and you repopulate back into the same eye, it has to be a self-sustaining thing. You cannot keep supplying limbal stem cells every now and then. So one has to find out what are the circumstances through which the limbal stem cell population was declining, or even if limbal stem cell population is there, it is not repopulating the corneal epithelium. One has to figure it out, otherwise it will be like giving parcel food every day. So one has to find out the root cause so that these newly put cells are doing the job what it has to be done in an ideal scenario. So if you see the human cornea in a cross section, as you can see in this picture, at the outer surface of the cornea there is an epithelial cell layer which sits on the basement membrane above the Bowman's layer. We will come to this structure. And the middle stromal layer, which is sparsely populated with keratinocytes and is surrounded by dense connective tissue. Remember, it is transparent. The final layer consists of a single sheet of endothelial cells which sits on basement's membrane. So you can see here that there is epithelial layer and basement layer, stromal cells and descent layer, and then you have got the endothelial cell layer, and each of them are marked here. Descent membrane is there, and basal cell is there, wing cell is there, squamous cell is there. And remember, the majority of this portion is transparent, as you can see in this structure, and that is essential for the light to pass through effectively. So homeostasis in corneal epithelium, the corneal integrity and therefore function depend upon the self-renewing properties of the corneal epithelium. We have mentioned this very clearly. The prevailing hypothesis is that the renewal of this corneal epithelium relies on a small population of putative stem cells located in the basal region of the limbus. Limbus is a structure, like I told you, between the cornea and the sclera, and in the basal region of the limbus there is a small population of cells which we call them as limbal stem cells. So these putative stem cells are primitive in nature, and they can divide symmetrically to self-renew and asymmetrically to produce daughter cells, that is transit amplifying cells, also known as TACs, that migrate centripetally to populate the basal layer of the corneal epithelium. So limbal cells have to divide in two different ways. One is a symmetric way to make a copy of themselves, or asymmetric way to one cell will keep copy of itself, the other one will become part of the cornea. That is called transient amplifying cells, these daughter cells. So the TAC, that is short form known as TAC, transient amplifying cells, the TAC divides and migrates superficially, progressively becoming more differentiated, eventually becoming postmitotic and terminally differentiated. Postmitotic cell means it is terminally differentiated, it has no plan of entering into the cell cycle, and many differentiated tissues have got terminally differentiated cells, and this is how the cornea establishment or dynamics is done. Let us quickly see the human limbus. So the limbal epithelial stem cells reside in the basal layer of the epithelium and marked as EP, as you can see here, which undulates at the limbus. You can see this portion is called limbus, and conjunctiva is there in the scleral layer, and the cornea is there, peripheral cornea and central cornea, and peripheral cornea, limbus and conjunctiva, and this is the area we are talking about. Daughter transient amplifying cells known as TACs divide and migrate towards the central cornea, that is mentioned in arrow. As you can see the movement here, which is migrating from here, arrow has been given, and to replenish the epithelium which rests in the Bowman's layer, BL, as it is marked here, and the stroma layer, that is ST, you can see here, and the stroma layer of the limbal epithelium cell niche is populated with fibroblasts and melanocytes and has a good blood supply. The stromal layer has the blood supply because they get the nutrition. Because of the stromal layer, the corneal epithelial cell is getting the nutrition. Otherwise, there is no blood cell. So how corneal cells will get oxygen and glucose? So they also have metabolism. So thanks to this underlying stromal layer. So this is the structure. It's a very simple structure, easy to understand. So this is your eyes black region, and this is your white region. So limbal epithelial stem cell. Throughout life, our self-renewing tissue rely upon populations of stem cells, progenitors, to replenish themselves following normal wear and tear or even after an injury. So the corneal epithelium on the eye's front surface is no exception. It also follows the same rule, as dead squamous cells are constantly sloughed from the corneal epithelium during blinking. You are closing your eye, you are creating a frictional force, and especially some people have got a situation called a dry eye where they put some lubricants. If dry eye situation is there, more and more wear and tear will be there, because wear and tear is less when you have enough of tears in your eye. So, but no matter whether tear is there or not, tears is there or not, when you blink, you are damaging the corneal epithelium. At the corneoscleral junction, an area known as the limbus, there is a population of limbal epithelial stem cell known as LESC, slightly different from LSC. LESC, limbal epithelial stem cell. Limbal epithelial stem cells share common features with other adult somatic stem cells, including its small size, high nuclear to cytoplasmic ratio, etc. They also lack the expression of differentiation markers such as cytokeratins 3 and 12, because they are stem cells. This LESC is also stem cells, so they don't have any differentiation marker, but they have the potential to give rise to the corneal epithelial cells. So this is an illustration of the stem cell control by the niche. It is thought that the LESC, limbal epithelial stem cells, undergo asymmetric cell division, producing a stem cell which remains in the stem cell niche to repopulate the existing stem cell pool, and a daughter cell that is transiently amplifying, which is migrating to the corneal layer, amplifying cell, that is called early transient amplifying cell, also known as ETAC. So this more differentiated ETAC is removed from the stem cell niche, and they can divide further. It will continue to divide, producing transient amplifying cell known as TAC. ETAC becomes TAC, eventually producing the terminally differentiated cells known as DC. So the double arrows, what you can see in this picture, arrows represent this self-renewing capability of the stem cell, that is recycling kind of arrow. The supporting niche cells, which is shown in blue in color, and they surround the stem cells, which are light green in color, light green and dark green, and DC cells are marked here, differentiated cells, and the tags are there, E-tags are there, and this is from the blue color stem are the supporting niche cells. So that's how they keep migrating. So to conclude this portion, that LESCs are important for vision, and efforts to specifically and prospectively identify these elusive cells are proving to be very difficult, because you do not have any bona fide or classified marker for these cells. Hence, one has to do a hit and try method. However, despite this, mixed populations of epithelial cells isolated from the limbal region can potentially restore the ocular surface and improve vision in patients with LESC function failure. That means if you have any problem with the limbal epithelium, you can collect the cells from the limbus and culture them, even if you don't know clearly what is LESCs clearly. To the mode of clinical efficacy, that is the treatment failure, may become an apparent once more thorough understanding of the normal LESC regulation, that is limbal epithelial stem cell regulation, and the role of the niche is gained. As of now, we do not have any bona fide marker available for them. The current approach is just like, in sometimes treatment, if you don't know the functional alkaloid, you take the whole tree, whole plant, and grind it and you consume it, assuming that that alkaloid is there. It's not a pure form. Something like that, researchers do that now. Limbal epithelial stem cells in corneal surface reconstruction. How does it contribute? The integrity of the corneal surface is essential for the ocular function. Injuries or congenital diseases could significantly disrupt the homeostasis of the ocular surface, especially the microenvironment of limbal epithelial stem cell. This will eventually cause the corneal regeneration dysfunction and decrease in limbal epithelial stem cell number. So the loss of LESCs for different reasons is named as limbal stem cell deficiency, or LSCD. Usually that is a medical term. One of the leading causes of vision loss worldwide, what we can see it. To restore the corneal surface, LESC transplantation in the form of tissue or cell culture is currently a viable alternative, a promising method to treat the LSCD. So limbal epithelial stem cell and their niche. We need to understand what is the niche, what is the importance of the niche. LESCs are a group of somatic cells that constantly asymmetrically divide, differentiate, and centripetally migrate towards the corneal center to replenish the worn out corneal epithelial cells. These corneal epithelial cells, CEPCs, known as the cornea's outermost layer, and they maintain the avascular and dehydrated feature because for the transparency for the cornea transparency and integrity, so that the clear vision can be obtained or maintained. LESCs are situated at the limbus, specifically at a transition zone of the cornea and conjunctiva, and that moves moves in a systematic manner of it is known as VOGT, walked. So this PV area, that is it is a Latin word, palisades walked. In short form, it is called PV area, is composed of radial fibrovascular ridges, and it is also accepted area for the limbal stem cell niche, or LSCN. Short form, it is called as limbal stem cell niche. So limbal basement membrane expresses a series of ECM proteins, laminin and collagen family proteins, including laminin 1, laminin 5, and type 4 collagen. Recently, many independent researchers in their studies have showed that the corneal regeneration could happen in the central corneas without involvement of the limbus. These phenomena indicate the central corneas could be the second niche itself of the LESCs. So several studies from animal models, which it's been done in rodents, that show that the cornea homeostasis could be maintained after artificial limbal destruction. You can damage it in the model organism, which indicates the existence of LESCs in the central part of the cornea itself. That means even if the limbus is damaged, sometimes cornea is not affected very badly because it has the stem cell niche in the cornea itself. So researchers suspect that the corneal epithelial regeneration in the central cornea could be the contribution of the transiently amplifying cell TAC migrated from the limbus some time ago, which is making a copy of itself and also maintaining. So limbal stem cell deficiency, if you look closely, LSCDs is defined as the irreversible impairment and dysfunction of LESC, limbal epithelial stem cell, and their essential supporting structure, the niche cells in the limbus. So without LESCs and LSCN, the cornea loses its regeneration ability towards the epithelium. Besides this, neovascularization and conjunctivitalization in the cornea can also be recognized as the feature of LSCD by simple microscopy in a pathological angle. But microscopy means you can put near the eye and then visualize it, which doctors will do. The diagnostic modality of LSCDs is currently poor and relies on clinical observation only. Impression cytology, which involves transferring, blotting, and immunostaining superficial ocular surface cells, is currently the best option for LSCD identification. You can put a layer and collect the cells and look at their health. That is how you identify. Limbal stem cell deficiency, if you look closely, LSEC's deficiency with different pathogenicity can happen due to various reasons. A is a healthy eye. B is a LSCD eye caused by chemical burn, as you can see here how it is vasculature has come. And C, LSCD eye caused by Stevens-Johnson syndrome, which will not go into the details of the disease. And D, LSCD's eye caused by herpes simplex virus. In all cases, it has become opaque or less transparent. So the clinical symptoms of LSCD, that is deficiency of limbal epithelial stem cell, observed from the patients include chronic ocular surface discomfort, vision loss, and photophobia. That means aversion to light. This causes the LSCD varies from individual to individual, and it can be categorized broadly into two groups: the extrinsic causes like chemical burn, radiation, infection like herpes simplex virus, keratinitis, etc. The intrinsic causes also there like Stevens-Johnson syndrome, aniridia, and chronic limbitis, etc. So cell transplantation therapy is one approach. The treatment of LSCD has gradually matured with the recent progress in surgical technique and regenerative medicine. To restore LESCs and LSN as a ultimate aim to LSCD treatment, regenerative medicine, which is defined by using somatic cells to regenerate the biological substitute and improve the tissue functions. This is the approach. You take the cells, have a scaffold and allow them to grow, and you make it. And it has become the most promising strategy for LSCD's management in the past 30 years or so. So cultivated limbal epithelial stem cell transplantation, known as CLET. So regenerative cell therapy using somatic stem cells has gained a great deal of attention in the treatment of multiple diseases in recent years. We know that. And a scientist, Pellegrini et al., first applied this CLET for the ocular disease. In this study, the autologous human corneal epithelium was co-cultured with the mouse fibroblast feeder layer and then transplanted into unilateral 2 LSCD patients. This is one approach. And in 2001, Coley et al. Established a method using human amniotic membrane as a substrate to facilitate cell expansion. This method uses feeder-free culture, no feeder layer needed, with no animal products. And this new technique successfully targeted or treated around 8 patients. So Mariappan et al. Also published a standard protocol for expansion of limbal stem cell epithelial cells in vitro in the same time, which is also a feeder-free technique using HAM, that is human amniotic membrane, as the culture substrate. So now CLET is one of the most mature and widely used stem cell therapies that have been reported. During the general procedure for CLET technique, a small limbal biopsy is performed to harvest healthy LESCs from the donor eye first. That is first step. Then the harvested LESCs are cultured and expanded in number in ex vivo. And this in vitro cells amount that is required for transplantation vary from patient to patient, and it carries such as amniotic membranes or other substrates will be simultaneously applied to support the transplanted CLET, because they need some transient support to start with. Depending upon the diverse LESC sources, this technique has been widely used in unilateral, partial or total LSCD scenario. So cell transplantation therapy is used for other approaches also. Although CLET performs well in LSCD treatment, the limitation of the source is very obvious. Every time you won't get enough cells. To overcome the shortage of LESCs, several recent studies have investigated the alternative sources of the transplantation. So the cultivated oral mucosal epithelial cells, known as COMET technique, using epithelial cells lining the mouth oral mucosa, and was first developed as a promising alternative to do the CLET. So in 2004, Nishida et al. First applied the transplantation of the cultured autologous oral mucosal cells to 4 bilateral LSCD patients. Bilateral means both sides. And all 4 patients showed obvious visual improvement. However, the long-term effect of the COMET shows a disadvantage, as neovascularization in the cornea was observed in all patients after transplantation, which severely affects the visual acuity. Inactivated mice fibroblasts were used as the culture substitute in this study, which also limits the future usage of COMET. Hair follicle derived stem cells are used. Human skin epithelial stem cells are used. PAC6 and Wnt7 also have been identified as a key molecule only for corneal epithelium differentiation, but not for dermal keratinitis. So to conclude, LESC's research has progressed greatly in recent years, but the development is still urgently needed many improvement. So significant progress in tissue transplantation therapies has also been achieved in the past 3 decades. In future, what we may find is a xeno-free culture can be made into existence, and new stem cell research and LSCD treatment has to be developed, and more effort should be put into this blooming research area to achieve more efficient therapeutic approach to conquer the troubles of a cornea fixation. We will study more about regeneration biology in the next class. Thank you.